Hello my dear students, welcome to the MS Science Academy. Myself Minu Srivastav, and today we are going to discuss about the topic of Unit 4, that is the sources of contamination. As we know, in the pharmaceutical industries, all the preparation have to be prepared in very much sterilized manner. Okay, a smallest contamination is destroyed our complete batch. Okay, so we have to be very cautious about that. Contamination will not at all allowed in our preparation. Okay, so first we have to see that what are the sources of the contamination, and in the next lecture we will see what are the methods of prevention for that we can stop these contaminations. Okay, now we will discuss about the contamination, that sources of contamination. Okay, there are different sources of contamination. Here I have represented the C for contamination. Okay, so different sources of contaminations are there, like atmospheric contamination, like raw material contamination, like HVAC induced contamination, like personal contamination, like equipment contamination, design contamination. One by one we will discuss all the points. Okay, first is the atmospheric contamination. What is the atmospheric contamination? In the atmospheric contamination, it's associated with the surrounding air. Okay, so in the when the atmospheric air has so many germs, because we know that in the atmospheric air we have so many germs, so many microbes, so many dust particles. So if that atmospheric air comes towards our sterilized place or comes towards our production area or the aseptic area, then it will contaminate our product. Okay, so how it will happen? Such condition can develop due to inadequate door lock system. If the locking of the door is not proper, that the air comes towards our aseptic area. Second is the improper sanitization. If in the aseptic area, if the sanitization is not properly done, then it will produce the contamination. Okay, and if the surface, floor are rough, so it will accommodate the organic material. Okay, organic material will deposit on that particular rough surfaces or the cracks, so it will produce the contamination. Second is that the raw material. As we know, the raw material is purchased on a bulk. Okay, so if the raw material will contaminate, then automatically our complete process will contaminate, or product will contaminate. Okay, so how the raw material contaminate? If inadequate storage conditions are happen, if the inadequate storage conditions are happen, then the material the raw material can contaminate. Okay, so the raw material degrade due to excessive environmental conditions like heat. If suppose that the raw material will expose to the so much heat, then maybe it will decompose our raw material. Maybe the raw material we which we buy, we just purchased, it will decompose with the due to the moisture. Moisture is the main source by which it will the moisture is the main source. It will contaminate or it will degrade our raw material. Okay, so if the moisture is present, then it will degrade our product. Okay, automatically. Now third is the personal. Personal is related with the unskilled or untrained persons which worked or which will manufactured or which will just supervising or performing the drug manufacturing. Okay, here the untrained, unskilled worker behind this, if they are not properly wearing that mask, gloves, so maybe their body skin some proteins, or maybe it will if their skin protein interact with the product, then it will contaminate our complete product. Okay, if they are not having the not properly having the proper hygiene, or if they are having some illness, so it will also, there will be chances to contaminate our product. Person who are supervising or performing drug manufacturing produce contamination due to improper handling and illness. Okay, so if in the personal cause, the improper handling or illness is the main cause, or untrained person or unskilled person are the main cause behind this reason. Okay, now fourth is the HVAC system. H stand for heat, V for ventilation, and AC represent for air conditioner. Here, heat, ventilation and air conditioning system So if the heat system, ventilation system and air conditioning system are not proper, then it will produce the contamination. Okay, so main reason, if what happened in the, if there's a air conditioner is present in that aseptic area, so it will organic matter if the organic matter accumulate in that air conditioner, so it will produce the contamination. Because if the proper because in the air conditioner proper cleaning, timely cleaning is essential. So if it will not takes place, then it will produce the it will contaminate our air, and automatically it will contaminate our product also. Okay, so here ineffectiveness of HEPA filter. As we know that HEPA filter is must in that mustly present for that sterilizing the air. Okay, so if the HEPA filter is not working properly or not cleaning regularly, timely, so it will contaminate our air, and automatically it will contaminate our product. Okay. Improper airflow. What is the proper airflow? The proper airflow means the airflow or the air pressure is more in the aseptic area. Air pressure is more in the aseptic area and it less towards outside of the aseptic area. So automatically air goes from high pressure to low pressure, and air moves towards from aseptic area to other room. Okay, so automatically air moves from that side, from aseptic area, and it will remove the germs also. But if due to that improper ventilation, maybe the air pressure will fluctuate, maybe the aseptic area has the low pressure, so automatically air comes towards the aseptic area. So if the air come towards the aseptic area, then it will proceed the germs, or it will contaminate, it may be contaminate our product. Okay, so here next is the equipments. Those equipments are or those containers are used for that manufacturing. If they have not having the proper design, if they are having improper design means they have any just groove or that other hole, so the material can deposit. And if they do not have the inadequate cleaning, they do not have the proper cleaning, so it will proceed the contamination. If maybe the cracks are present, CRSC case, if maybe the cracks are present in that container or that particular equipment, so it will initiate to deposit the organic material, and that if the organic material deposit, then it will just contaminate our product. Improper sterilization takes place. If in the container system not proper sterilization takes place, then it will proceed the contamination. The last is the design. What is the design? As in the aseptic area, the walls having the crack, walls having the rough surface, and the walls having the cracks on the floor, and that cracks, that rough surface and the crack on the wall will proceed or will lead to accumulate the organic material deposition. So it will proceed the accumulation of organic material, and automatically it proceed the germ contamination. It will proceed the contamination. Okay, accumulation of material on the cracks. Okay, so it will initiate the germination, it will contamination. Okay, if the ceiling is not cleaning properly, if the ceiling is if the ceiling is at too much height, okay, so the cleaning, if it's not properly done, then it will proceed the contamination. In this lecture, we have discussed about the sources of contamination. In the next lecture, we will discuss about the methods of prevention of contamination. Hope you enjoyed the lecture. Any doubt, you can ask me in the comment section. Otherwise, please like, share and subscribe. Thank you.